string looks like something like this. This pattern content basically. And the other one looks like the opposite of this. That is, you have a pentagon, but it's upside down.
Right.
Does it make
And
sense? This is what actually is meant by. Yeah. U3. And you have bases here, whatever bases there. So like A, T, G, C, A, T. So you will have T. Similarly, you will have. So look at what ends are actually free here. 5 prime is free here, and 3 prime is free here. Correct. And as I told you, like here, what is free here? 3 prime is free, right? 5 prime is towards the bottom in this case. So this is the true reason for the antisense. Like it is literally antisense because the ends are opposite. Like it's put exactly in the opposite manner. Now let's talk about the kind of. Replication happened from 5 prime to 3 prime with you?
That is 3 to 5, right? I don't remember, but I remember 3 to 5.
It's all, anything that is ever made is always made in 5 prime to 3 prime direction.
No, but there was something about 3 to 5, right?
Yeah, it moves along the replication fork in 3 to 5 direction. And इसका कहानी अलग आएगा. 3 to 5 वाले का कहानी थोड़ा complicated है. I'll come to that.
Okay.
So like it's not as direct as you think it to be. Cool. Like for the time being, let's not discuss that अभी के लिए. I'll come to that. Yeah.
Cool.
अभी दो मिनट दे दो, मैं आ जाऊंगा उसके अंदर. So our DNA structure is basically 2 strings. I'm now ignoring this helix. The helical structure is because of how like actually you can see that I cannot, if I add a P here, it should go inside, right? इसी स्ट्रिंग को मैं अगर उल्टा करूंगा तो X शेप का बन जाएगा. One second, like this string that you're seeing, right? ATGC wala.
Right.
The string I made, DNA string. So if I sort of, one second, where is my lasso tool? Never mind. Yeah. Okay, like it's I mean it should sort of move away. Like इसका natural tilt इस तरफ है. Its natural tilt is towards the other side actually. If you're able to see, like it's moving apart.
Yeah.
But it's not actually moving apart. It's actually something like this crossed over. So it's actually twisting around each other.
Right.
So here, this thing is moving to this thing is moving to this side, and this thing seems to be moving to the other side. But that's not what is happening structurally. It's actually coiling around each other. So this can be thought of as ऐसे आ रहा है. अभी दूर जाएगा, थोड़ी देर बाद पास आएगा. It coils around each other like that.
Yeah.
You can see it, right? Yeah.
Yeah, it makes sense. Yeah.
That is how the structure looks like. And let's forget, for imagining that for a second that they're linear strands, let's look at how the bonding works. So your A corresponds to T. T would be flipped. You understand why I'm saying flipped, right? Yeah. So like T flipped को मैं अभी के लिए T मान रहा हूँ, but T is flipped. Imagine it as, you see it as flipped. Okay.
Yeah.
G will be with C. And the stand doesn't matter. This rule remains the same.
Yeah.
Irrespective of stand. Like A के सामने T होगा, T के सामने G होगा. Why is this? So there's something known as purines and pyrimidines, right? Pyrimidines. You remember this?
Yeah, we
Intro to
remember
bio.
this.
Quick question.
Yeah.
So, what is a purine? Which one of them are purines and which one of them are pyrimidines?
I will tell you. The purines were the bigger ones and pyrimidines are the smaller ones, right?
Yes.
Okay, that
Purines
much
have
I
two
got.
cycles. Uh, two cycles. So basically, they have general structure which looks like. I mean, I'm talking about the general structure. The precise structure is slightly different. So you have one aromatic chain, like there are small internal differences, but essentially they both are something like this. Okay, I'll just make the entire thing. So this is one of the pyrimidines, sorry, purines, and the other would be the same thing with N. That is the only difference. This group is called, this double cycle group is called pyrimidine.
Purine.
Sorry, purine.
Right.
My bad.
Huh.
Purine. Oh God, I'm so dumb. I'm so sorry. Yeah,
So it's fine. It's
I'm
confusing as it is.
confused between. So my question was not that. Like, my question was which are the pyrimidines and which are the purines.
Look, anything that ends with INE is a purine, right? Pyrimidine ends a little different. So adenine, guanine, cytosine. Oh damn, everything ends with INE. I don't remember.
Yeah.
Yeah.
Just try to recollect. Meanwhile, I'll show you the structure of pyrimidine also while we are here. Pyrimidine is also 6 numbered, except that it has. So here it could be N group or double bond O group. So I'm leaving this and I'm writing some F. Cool. Similarly, here you can have F here, functional group here. Cool. The purines are guanine and adenosine. Pyrimidines are cytosine and thymine.
Right. Let's see.
So,
I completely forgot.
yeah. So I thought you might have, that's why I asked you. Yeah. Like this is important, like what is purine and what is pyrimidine. And that's a reason why they are alternating also, right? Do you remember the reason?
Which one? Alternating as in why A bonds with T.
Yeah, why purines bond with pyrimidines?
So this I remember for sure is because of hydrogen bonding. And that has something to
Hydrogen
do
bonding
with...
is happening, but like why do purines bond with pyrimidines and not purines bonding with purines?
Oh, okay, that way. Because it's spatial feature and the size or something. I don't know.
Yes, the size. This is double, like double sort of double cyclic, right? This is single cyclic. So purines are longer than pyrimidines. Purines are comparable sizes with purines, and pyrimidines are comparable sizes with pyrimidines because they have one cycle only, right? Functional group is not that important.
Right.
It's just one atom. So you have to maintain the distance between DNA is equal, right?
Yeah.
This distance should not be greater at some places and smaller at some places. So
Yeah.
that is why this alternating structure is there. Like pyrimidine always binding with purine, so that the distance remains the same.
Mm-hmm.
Now about hydrogen bonding, A and T have 2 hydrogen bonds between them. G and C
G
have
and
3.
C have 3. Yeah.
Yeah. So this is how hydrogen bonding is there in these in the DNA. So so hydrogen bonding, as we know from chemistry, it's a pretty strong bond, but like it's not strong enough like ionic. It's a dipole interaction, right?
Right.
Dipole interactions are weak, right?
Yeah, it's not as strong as ionic or covalent.
Ionic or covalent bonds. It's not actual. The bonding is very weak, right?
Right.
So this gives DNA the property that it can split into 2 strands easily when heated.
Yeah, that's denaturization. Right. Or is that for protein?
That is denaturation of DNA.
Okay.
But like once you heat it and you leave it, it bonds back again because this ATGC thing sort of this complementary nature is there. So it sort of comes back together. Assuming that you do not unravel the entire thing, then it won't. Then it will sort of fold within itself, which is not good.
Right.
Yeah. That's how DNA is destroyed, right? If you like people sterilize stuff, right? How does sterilization work? You would have seen doctors and all, they put their knives and everything in ovens, right? They'll put their surgical knives and all in ovens and they'll heat it. So what it does is that heat denaturizes proteins and DNA. So proteins is more important for bacteria and so on. DNA is more important for viruses and so on. Like
so
viruses can survive.
technically, if you cook food, then the protein that you're eating is denaturized.
Yes. I'll give you an example. Take egg. What happens to an egg once you boil it? Does it still look like the same?
Kind of, it solidifies and kind of become a big lump.
Yeah.
Of liquid.
Yes. That is denaturization right there.
So
Its nature was something
eat food, is it better to eat uncooked stuff because you're technically taking in the protein as it is? Like assuming
Not
it's
really.
clean and all that stuff.
Not really. It depends on your digestive system, to be honest. Some things are better cooked. So the only case against cooking is actually this, that some of the vitamins and essential nutrients, they evaporate off when you cook, especially when you cook in an open manner. So along with the water, they just float. Like you end up not consuming it at all. That is the only case to be made against food. And second is that fibers are sort of weakened. Other than that, food is actually better for digestion. Why is it cooked food is better for digestion? The reason is that when you're denaturizing it, denaturization is basically breaking of bonds, right? Now, mostly denaturization will change shapes and all, but it will also break bonds here and there, which will make the body's job of breaking bonds easier because some of the work has been done for it.
I see. Okay, so wait, if you heat something and assuming it didn't lose most of its structure and it cools back down, then it's possible that those bonds form back. That's why you're supposed to eat like warm food.
No. That bonds won't form back. Warm food का concept is different. Like warm food is because heat. That is because of the second advantage of cooking. When you heat stuff, it's a sterilization process. You're killing bacteria and other stuff that is there. For example, cabbage. Okay, this doesn't look like a cabbage. Now it looks more like a cabbage, right? Cabbage has this layered leaf structure, right? What happens is that worms, including tapeworms, they lay eggs in these layered leaves. So uncooked cabbage is actually very bad. You can end up getting worms in your stomach. So cooking actually helps it. Similarly, like if you eat meat like pork, the pigs are grown in a muddy environment, right? And like the earth generally has worms in it again. So pigs are likely to be infected by like tapeworm, like a tapeworm or liver fluke and other such worms. So they'll have eggs all over like the pig's stomach and inside the meat. So that's why you have to cook pig meat at like pork at very high temperatures. So they cook it properly.
Like what I was thinking was, if you heat something and then you let it cool, would those bonds form back in food?
They won't, right? If you cook an egg, will it ever become back the egg? No, it will go back, but it won't ever become back the egg. That is what.
I mean, you're talking about DNA. As long as it doesn't unravel itself, like it can form the bonds back. If you remove the heat,
DNA can form the bonds back. Like DNA is extremely, how do I put it, resilient. It's very hard to destroy DNA. One of the only good ways to destroy DNA is to put extreme ultraviolet radiation on it. And I'm talking like pretty high amounts of ultraviolet. Something similar to your solar flare.
You
know solar flare, right? So So solar flare immediately causes skin cancer. What is skin cancer? It is destruction of your DNA because of
DNA.
high UV radiation.
I see. Interesting.
So it requires high amounts of ultraviolet to actually destroy it. DNA is very resilient. You can't break it just like that. In fact, for cooked food and all, DNA will be intact. Nothing will break there. So, but in this process, you will be killing what? Bacteria. Most of the viruses, most fungus, eggs of various pathogens. Like this includes worms, other sort of parasites.
Right.
You can have worms, you can have flukes, and so on. These are different things that can come through the food. Now, why do they say that have your food while it's warm? Because more time you let it cool, then more chances of bacteria and fungi reinfecting it.
Right, that is there. And the other thing is, they do say that it helps in digestion if it's warm. So I presume that is because of the bonds kind of breaking, and when you let it cool down, I thought if the bonds form back, at least in some sense, it's harder to break. Right. So you'll be doing more
Okay,
work.
I'll give you a case against this digestion aspect of it. Like cooked food is good for digestion, but warm food may not be good for digestion. May or may not be. Like take case of apple or banana. It's not warm, right?
Right.
Is ice cream harder to digest than tea? It's not, right? The thing is that we are accustomed to eating hot food, so we prefer hot food. It also helps us maintain our body temperature. We feel better with hot food. And that's also because our body temperature is also 35. If it's too lower than that,
okay,
like
it might be that
within our body.
might be that the enzymes you release is at some particular temperature.
Yeah, exactly. It has that optimal temperature that we learned in ITB.
Right. Makes sense.
So, like, actually, truthfully speaking, room temperature food is the best. But it has highest chances of contamination. So, hot food. That's why you have hot food.
I see.
So, one more question: like, when you put your food in fridge, you reheat it, right? So, why do we reheat it? We don't need to cook it, right? So, it's again this question of bacteria and viruses.
Yeah, I mean, though they'll still be there, but they might be dormant in the when it's in the fridge. So their rate is lowered on decay.
Their activity is lowered, right? Because in low temperature, we learned how biological activity slows down.
Yeah. So you can just preserve it for longer, but you still have to reheat.
You have to reheat to kill the bacteria, so that you don't end up with some disease or something.
Right.
So reheating is more about sort of deleting the contamination that happened because of the food cooling down and being kept out in the open.
I see.
So anyway, that we went to a divergent. The reason why I said that this DNA unravels is because it has lots of implication for PCR, a polymerase chain reaction. So, like, unless you provide extreme amounts of heat, I said that unless the DNA doesn't completely unravel, it will still come back to there, right?
Right.
And now I'll put a case why DNA cannot unravel completely. At least let's consider our DNA. We have 30 billion base pairs. 3.2 billion base pairs. Do you think we can?
3 billion.
Yeah, 3 billion. Do you think you can unravel ever? Like, so many base pairs. It will take forever to do that.
The reason it's so condensed is for the amount of space it takes up as well. Right? If you try to unravel the entire thing, it's just going to take too much space.
Space is light. Like, if suppose you had infinite space, the amount of temperature and amount of time it takes to unravel is huge.
Right.
So, like, that is one advantage of DNA, right? Like, DNA survives with very little damage. So, actually, this is my research. Like, I'm studying on, because DNA is very hard to destroy, I'm studying on how to store data in DNA so that data won't be destroyed.
Oh, interesting. Okay.
Yeah.
So you mean something like you code up some sequences and try to impart that into the DNA so that it keeps getting replicated and stored in all the future versions of that organism. Something of that sort.
We are not putting it in the organism itself, bro. Like what we are doing is that we are printing out DNA in the same way
Hmm.
you would print out any other in paper you would have print out. Instead of that, you are just causing this reaction to happen, like polymerase reaction to happen. You have a DNA
Hmm.
strand and you have the complementary strand. You have a DNA
Hmm.
molecule, right? This will store in a sort of micropore, which is set like sort of like a cup. You store it in the cup, and now what you do, whenever you need it, you sort of get it back. That's it. So from binary,
Okay.
you make DNA. DNA, you get you store it, and then you get back whatever DNA you had stored, and then you bring back binary. That's it.
Okay. Okay. So you're not doing anything new. You're not manipulating DNA. You're just using it as a medium to store.
Store, yes,
Okay. Okay. I see. So
because
also.
DNA is very good at storage. It
Ah.
cannot.
Okay.
It's
Okay.
very susceptible not to have damages. It's very hard to
Mm-hmm.
damage DNA.
Right, and every storage device you have has a lifetime, right?
Yeah.
Like hard disk will have say 10 years or some kind of lifespan.
Meanwhile, DNA stays for millions. So GG.
Right. Okay. Interesting.
Yes. Anyway, so where were we on the structure? Like it has hydrogen bonds which break upon heating.
Hmm.
Cool. Yeah. I tend to go on off topic, so like you just
So no, it's
tell me
fine.
when I'm
Off-topic
going
discussion
apart.
is very fun.
Shreyas, are you there by the way? Yeah, I mean I didn't hear anything from you, so I just thought I'll ask. I sound like a teacher now.
It's
Oh no, 1 second. Where was I? Yeah. Uh, what was I talking about? I forgot, sort of.
so you're talking about in
Double bond,
PCR.
hydrogen bonds in DNA.
Yeah, in PCR, why
Like
it's
the
kind of
heat,
useful that it cannot completely unravel it. So.
it's useful that DNA strands can be separated out by heating. That is what's useful in picture. So if you have a DNA strand, you apply heat, it will uncoil. You remove heat, it will recoil up to certain temperatures. Like
Right.
you can't just arbitrarily heat it to sun's temperature or something and expect DNA to survive. ठीक है, like around like in most cases it's around 100 degree 200 degrees
Okay.
and that's it. Yeah, it's around 100 degrees. So this property that it can split and come together is very important for life itself, right? That was what I was saying. It's very resilient. Sort of come back,
Right. Right.
whatever it.
But also
Yeah.
our DNA, right? Our human DNA, is it present in like every single cell that there is?
Yeah, every single cell.
So basically, if you just take a swab and
Except
take out
actually your
RBCs, right?
this thing,
Except
RBCs.
RBC.
Yeah.
Yeah, except for
Yes,
them,
you
everything is
remember,
good.
nice.
Yeah.
Nucleus in here.
So technically, you
Actually,
can do a DNA match between like a parent and child with just saliva and stuff too, right? That's how it's done.
so saliva by itself doesn't really contain cells.
But
It's
cheek
only
cells.
when you say cheek cells. When you have cheek cells, you can
Cheek cells.
contain.
Yeah, yeah. Not saliva. Cheek cells.
So actually, we did this in school. Like we used to scrape out our cheek cells, and we
Yeah.
had to like we didn't have DNA testing. We had we had to scrape out cheek cells, and we had to observe them in cells like in microscopes and all.
No, even we had
No,
cheat
like
cells,
this
and
is
that's why I remember.
NCRT wala thing.
Yeah, NCRT thing.
Yeah.
No, 10th grade
Uh-huh.
this was. 11th and 12th we didn't have it.
नहीं,
9th or
10th
10th.
में भी था, 11th and 12th में भी था. Animal, there was tissues वाला chapter, right? 9th or 10th में. I'm not sure.
Oh yeah, I mean, I
There
didn't
is a
take bio
chapter
in
on
9th,
animal tissues and
in
plant tissues.
11th, 12th, so I didn't do it then. But 9th and 10th, one of these years I definitely did.
Yeah, 9th and 10th में भी था.
Tissues
Yeah,
was
right.
there. Yeah, tissues chapter.
Tissues. Yeah. Tissues chapter में. So we had animal tissues, plant tissues, whatnot. Yeah. Anyway,
हाँ.
heat करने से it uncoils, and when you remove heat, it recoils back as it is to certain amounts of heat. For all practical purposes, assume that it recoils back.
Okay.
So, yeah, that is basically about DNA. Now we'll talk about replication, which is how I started this entire process. So, you have this coiled strand. How do you go about replicating it? First of all, first thing that you would have realized is that to replicate it, one unique challenge is that you have to replicate both strings. You can't just replicate one and be done with it, right? Because there are two strings in this.
Yeah.
So, 1 second. Oh. Correct,
There are
correct,
some
correct.
two ways of doing it, right? Like one way you do it, you get like another complete copy, so it explodes exponentially in DNA replication.
I'm sorry.
So like every time you take two strands, right? Like this is a two strands of like DNA has
Huh.
two strands. So you replicate each strand and then you have like two copies of DNA. So you those two copies, if you continue, then you'll have four copies, then eight copies. So it'll grow exponentially, right?
Yeah. But like that is actually again in PCR. Like I think I was trying to talk about semi-conservative thing that whenever a new strand is made, one of the strands of the newer DNA would be from the original DNA, and one of them would be new recreated.
Yeah. Yeah, this is what you told
So
me. Yeah, I remember.
yeah. So like one original strand will go to each of the new children.
Right.
But even before we come there, like what happens is that there is a protein called helicase. DNA.
Yeah, exists.
Yes. Yeah. So like the helicase doesn't arbitrarily join anywhere. There are certain regions called origins of replications. Origin of replication. This is also known as Ori site. How creative,
I
right?
remember this
Origin
name.
का Ori निकाल दिया. Right. So Ori sites are basically origins of replication. So what happens is that our poor helicase, it joins, it attaches to the Ori site, and what it does is that essentially it causes the DNA to sort of split into two parts. Now the question is that I've made it somewhere in middle. So what happens to stuff that is below the Ori site? The answer is that it looks something like this actually. But for all practical purposes, we can think that it's a start over. It's easy to understand. Okay. It's actually the DNA is unraveled below, but we don't need to care about the DNA, whatever is below here. Replication is only happening in this direction. So now what happens is that our helicase has uncoiled it for us. Okay, so the first step was uncoiling. Coiling. Now another enzyme comes to play. Now this is the true worker of this process. It's called DNA polymerase.
Right, I remember this.
Now, this DNA polymerase, there are multiple types of DNA polymerase. This type of DNA polymerase is called DNA dependent DNA polymerase. Cool. Because it depends on the DNA. Okay, we'll come to DNA dependence later. It's a DNA polymerase. Cool. So what DNA polymerase does is that enzyme helicase has led to uncoiling of one end of the thing, causing this fork. This fork is called replication fork. Intro to bio again. In replication fork, the DNA polymerase would come and it would start making DNA strands here. Now this is 3 prime, 3 prime and 5 prime. Now, remember from whatever I've taught before, DNA can only be produced in 5 prime to 3 prime manner.
Yes.
So, अब DNA polymerase जब चलेगा, it will move in this direction. Correct. It is moving in one direction only, right? हाँ. So, यहाँ पर 5 prime to 3 prime बन रहा है. Correct. Now the question is, यहाँ पर तो नहीं बनेगा, because यहाँ पर you require a 3 prime to 5 prime sort of structure. So what happens is that it does something very smart. It makes 5 prime to 3 prime for a short step, it goes back up, 5 prime to 3 prime for a short step, goes back up, 5 prime to 3 prime for a short step. It does it in a staggered manner. It does. So the direction of replication, like production, is still 5 prime to 3 prime. 5 prime से start किया, 3 prime तक बनाया. Then it makes another fragment which is from 5 prime to 3 prime, and so on. It cannot make one stretch like this, so it makes it into smaller chunks.
Right.
So this is called staggered replication.
Yeah, I think you taught us this as well. Now that you mentioned it in pieces, I remember it.
Yeah, and these pieces are called Okazaki fragments.
Right, we had this in one of the ITB quiz.
ITBs, yeah, fragments. Okazaki fragments are literally in fragments. You have to join them together. So, this entire thing is joined together by what is known as DNA ligase.
Ligate, yeah, I remember.
So ligase comes from the word ligate. Ligate means to join. So it basically joins this stuff up. So you have these staggered fragments, right? ये अभी ऐसा है. After some time, it will be somewhere on top, right? I should have used different color for the new strand. So ligase will join this up, and I should have used a different color. So such a dumb teacher.
I mean, you literally taught us more than what we learned in all these years.
So after some time, your polymerase should be somewhere up here. Ligase has joined this. So essentially, this is what it will look like.
I see. So yeah, so while DNA polymerase makes the string in 5 prime to 3 prime direction, but since our frame of reference is usually 5 prime to 3 prime in the original DNA strand, it looks as if it's moving from the 3 prime direction to 5 prime direction, right? Like that is where that directionality seems a little weird. That's the reason.
Oh no. Okay, so it's actually kind of the opposite. The new strand is being produced in 5 prime to 3 prime. It is reading the DNA in 3 prime to 5 prime.
Correct. So it's going from like 3 prime to the 5 prime direction, right? Like it's moving in, starts from 3 prime of like the left string and then moves towards the 5 prime of the left string in that picture.
Yeah.
So
So
while synthesizing, it's
with respect to
technically
the original
synthesizing
string,
from 5 to 3. Yeah,
yeah, the new
I see.
string which is being synthesized from 5 to 3 in both directions.
Right,
The
right,
old
right.
string is being synthesized. यहाँ पर break करके हो रहा है, but still the production is always from 5 prime to 3 prime.
Right, right, got it. Huh.
But like with respect to the original string, 8 के respect में it is doing from 5 prime to 3 prime, and with respect to the other, it's 3 prime to 5 prime.
Yeah, got it. Huh.
So that doesn't really matter. Like only thing that matters is that
Yeah, production of the
what direction it is
DNA
producing the DNA.
is right. Yeah, got it.
Keep this order in your mind, like 5 prime to 3 prime. Everywhere you'll see the same order. And this is sort of like universal. And the reason for universality I explained, right? That's because we talked about polymerization, tail polymerization, head polymerization during ITB.
Yeah. Which direction it comes in. So
Yeah,
it has to
this
attach
is
to
the
the
tail.
OH end. So
Yeah, this is a tail polymerization, right?
right. The head
So
is considered as the phosphate group. Right?
yeah. So you always, it sort of attaches to the tail, like whatever is the string, the molecule comes and attaches to the back. So that is why this thing is from 5 prime to 3 prime. Cool. So this is what it will eventually look like. Your fragments, like after ligation, this will start unraveling, like coiling with itself. And how does the DNA? This DNA is corresponding to the other strand, right? That's why, like, how it's replicated. Correct. If you remember our, what did I do? I had wanted to paste this. So this was what. Don't mind me. I'm messing stuff up. So this is how our final picture should look like, right?
Right.
And you are seeing how it is happening. And I mean, there will be base complementarity that is formed between new and old. So यहाँ पर A था, तो इसके according यहाँ पर T था. In other words, what is being formed is exactly same as ये वाला strand. Yeah. ये वाला strand is exactly same as ये वाला strand. Strand same as this. Strand same as this. Essentially, you end up getting 2 copies of the DNA.
Right.
So now this DNA polymerase is polymerizing based on what was there in the DNA, right? It looks at what was in the DNA, and then it adds the nucleotide which is corresponding to it, complementary to it.
Right.
So it depends on the DNA strand, right?
Yeah.
It polymerizes based on what is in the DNA strand. So it is called DNA dependent DNA polymerase.
Okay, that's the dependency.
Yeah, because it's literally reading from the DNA and like polymerizing according to what is complement to what it's reading.
Makes sense.
Yeah, so that's how replication works. And replication works in this semi-conservative fashion, that is, there is an old string and a new string together. ITB stuff. Now we have done replication completely. Woohoo. Now we come to see this time around it went much more easier because you remembered stuff.
Yeah, we kind of remember a lot.
Yeah, because I remember उस समय मुझे replication fork में काफी time लग रहा था and like yes, so GG. So replication is over. Now we have transcription.
So wait, in the replication procedure, what was the application? Was that complementary DNA and PCR stuff? No, like whatever she taught.
Replication.
No, whatever she taught was the application, the PCR and I
Yeah,
don't know
replication
about CDN.
comes in PCR. It comes in PCR. I'll come to that. I'll come to that. Like replication as well as this heat component comes in PCR. So like once I set the base, then I want to teach all of this, and then I'll go do the basics, and then I'll go to the biotech one part, which is completely new, I suppose.
So wait, I remember transcription kind of well because you taught this also. So wait, in transcription, basically whatever DNA is there is copied in the exact same strand, and but in terms of RNA, that information is stored. And then I think there was something about mRNA and tRNA, some types of RNAs in this. And then finally, when
Yes.
transcription comes, it's like is one huge protein, and then it reads three at a time in that whole procedure that that we just coded up until it comes to the start codon and makes a chain, and then kind of cuts off the chain when the stop codon comes. Right?
Yeah, sort of correct. Yeah, mostly correct actually. So,
Yeah, I mean, I remember the
so,
concept. I don't remember details.
so one thing in transcription is that there are two strands of DNA. Correct. Which will you transcribe from?
Again, 5 prime to 3. Interesting.
But just think of it. ये मैंने 5 prime to 3 prime लिखा है इस डायरेक्शन में. मैं इसे उल्टा कर दूंगा. मतलब उसे कैसे पता कौन सा ऊपर है कौन सा नीचे? नहीं, रैंडम नहीं होता.
Interesting.
I'll give you a clue. Like, how did we? Correct. Kind of. Remember, there are ORI sites here,
Right.
where like helicase would come and it would join. Something similar. Helicase also comes here. It sort of opens up DNA, and your this process is very similar to transcription actually, except no. ORI site पे नहीं आएगा. Like it's a different kind of helicase altogether. So the site where it binds is called initiator site. Let's first talk about why we don't have like let's talk in a very like logical sense. Yeah, you know that one is transcribing the other, right? Why wouldn't we want to sort of make RNA for both? हमें क्या फर्क पड़ता है? Why don't we make for both? What is the
I don't
disadvantage
think
if we make
we,
both?
yeah, I don't think we need it for both because just a duplicacy in terms of information, right? And then
It's
you
not
want to
right.
generate a protein.
For example, if your this string was ATGC,
Huh.
this string was going to be what? This is 5 prime to 3 prime. It is going to be G C A T, right? 5 prime to 3 prime. Does it make sense?
Yeah, I got it. There is no duplicacy. Yes.
There is no duplicacy. Unless it's a palindromic syndrome, there is no duplicacy.
Yeah, there
Unless
is
it's
no
a
duplicacy. But it's just that
unless
it's
it's a
obtainable given one strand, but it's not
yeah,
duplicacy.
it's not duplicate. It is complement sign. Reverse complement sign. To be more precise.
Yeah.
अगर मैं
It should
इस
be.
स्टार्ट should be right. ये दोनों आर कॉम्प्लीमेंट डीएनए. इसे मैं एक साथ छोड़ूंगा तो इट विल डू समथिंग लाइक ATGC and GCAT. I should write this in ulta.
Yeah. I am right now because even I assumed it was duplicacy, and it doesn't matter which one we took, but now it
See,
makes sense.
this is how you're seeing it, right?
Yeah,
Casting literally ऐसा है. GATC and ulta GCAT. But in essence, this is actually GCAT, right? This is not ATGC.
Yeah, you still code for
even
different.
complement देने से duplicate होता है. Yeah. The complementarity sort of actually reverse and complementarity both handle this, right? That it is not duplicate unless you have a palindromic syndrome. So you would have noticed that palindromic palindromic syndrome क्या ना sequence palindromic sequence तुमने notice किया कि biological thing में it's not really a palindrome because G A A T T C exactly when you do इसका reverse complement that is इसका corresponding thing it is G A A T C. So like to get it back again I think this flipping thing works a lot, like it clears your mind about how ideas work. G triple bond, double bond, double bond, double bond, double bond, triple bond. This is how the actual DNA was said.
Yeah.
That is why it's palindrome, because you are reading from 5 prime here. जब उल्टा read कर रहे थे तुम यहाँ से 5 prime पढ़ रहे थे.
Yeah.
GATTC, GATTC, both sides. You don't read from 3 prime to 5 prime ever.
Yeah.
Yeah. So question is, how do you decide? Okay, my question was why don't we make 2 RNAs? The proteins are obviously different. So why don't we get?
Yeah. Why don't we make now that you get two different sequences altogether? I was under the impression that there'll be some duplicacy, so we didn't have to do it. But now that I think about it, it's not going to be that. Exactly.
The thing is, if something is coding for a protein, like suppose this sequence codes for a protein. Suppose I wanted methionine, leucosine, glycine, or that is what my body wanted. So it will be ATG, leucosine is AAA, glycine is GGG, and yeah, so on. This is what this chunk is coding for. I have no certainty what my opposite chunk is coding for. It will code for some random protein which I don't want.
Right.
It's a wasteful process, so why should I even do it?
Yeah, but so how do you know which one to pick?
Picking is decided by initiator site.
So that is okay. So does the site pick which point to start from or which strand to do?
Which strand and which point?
Oh, I see.
It's like an origin of replication, except that your strand is also picked.
Okay.
Yeah, this is a lie. This reason is a lie. The real reason is even more mind blowing. Suppose you made RNA for both. तुम्हारा एक RNA बना, इसका corresponding तो ये होगा A U G blah 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 from. Actually, I should have made it ulta. My bad, because it's going to be read in the opposite direction. Right? Does it make sense? Okay, like let's not let's ignore this. Numbering volume when I discuss something. Suppose my RNA from top one, considering top one to be template, AUG so on. नीचे वाले को template मानूंगा. This will be from 5 prime to 3 prime. मान लो. नीचे वाले को मानूंगा. It will create something from 5 prime to 3 prime. But its last thing will be it will always be complementary to this, right? C. U. So I'll give you an example. ATG. Let alone protein coding वाला thing. I'm arbitrary sequence ले रहा हूँ. ATGC, ATGC, ATGC. Cool. इसके corresponding for a second मान लो complementarily बनेगा ना. This is 5 prime to 3 prime DNA. इसके corresponding DNA क्या होगा? TAC, TAC, T.
No, wait 1 second. Yeah. No, you mess. It is instead of T, it is G, and A places T.
G.
Right.
TAGC, sorry.
T A C
TACG.
A G T A G.
TACG.
Yeah.
Yeah. This is the two DNAs, correct? And this will be from 3' to 5'.
Hmm.
So what will be the DNA, RNA corresponding to this? This will give you U. इसके corresponding, हाँ ऊपर वाले के हिसाब से क्या? UA. हाँ.
Yeah.
Good, like good observation. अब next वाला इसका क्या होगा? Same, right?
Right.
Do you see the issue now?
When sitting
These are
upstairs, thank you
complementary.
from. Right. That's a good
Won't
thing,
it
no?
form double
Okay,
stranded RNA?
okay. I see what happened. I see what
Won't it
happened.
form double stranded RNA? Because it's complement to each other, it will form double stranded RNA. And double stranded RNA is useless because तुम्हें unravel करना पड़ेगा protein बनाने के लिए. And unraveling is an energetically heavy process. It's very hard to keep them unraveled. So you don't want it to ravel at all. So what do?
Yeah, when you asked me
You
if
don't make
something
the
was
second
wrong,
strand.
I was like, that looks even better. It looks like DNA, but with DNA replaced.
So you don't make the other one at all. बेकार का झंझट नहीं करना. We don't want to make waste proteins. We don't want to separate it all. Yeah, that is the reason. So the reason.
I see.
So initially, when life started, we were all RNA. In fact, we were all double-stranded RNA, which would form single-stranded RNA. And single-stranded RNA will make proteins. That is how it was. So this is called RNA world hypothesis. All of this was RNA. This is how early life was. But then, so you had double-stranded RNA, which will form single-stranded RNA, and then proteins. Cool. Then,
But this was inefficient.
This is inefficient because RNA is very unstable. Because of what?
Okay.
Your extra OH, right?
Yeah.
Because of that extra OH, it's very like this thing. How do I put it? It is very reactive. So,
Right, you said it'll get oxidized and then it'll form H2O and then the base would get oxidized.
yes, it will oxidize. It won't get oxidized. It will get oxidized. It will get. It will oxidize, right? It's an OH group.
Right.
Yeah. It will get reduced. Like RNA would get reduced. It's a highly oxidizing thing, RNA. So it was unstable. So even with double-stranded RNA, it was unstable. So we went to double-stranded DNA to make single-stranded RNA and then proteins. So its advantage क्या हुआ? We got a much more stable storage medium. But look at this, both cases. Like whatever produces protein has to be single-stranded. You can't make double-stranded, like proteins with double-stranded stuff.
Right.
So that is the crux of the story I'm telling you. Like the reason why it doesn't produce both from both is because you don't want double-stranded RNA to come. फिर तुम protein नहीं बना पाओगे. Because unraveling नहीं होगा.
Yeah.
Yeah. So that is done. So on the basis of which which DNA is being replicated, the DNA can be thought of as like template or like coding strands. So suppose the initiator site is on the DNA on top. Okay. This is the template for RNA formation, right? Your like what do you call, your polymerase would attach here, and it will make RNA thing based on this. Actually, I should have put it from the other end, right? F for me, bro. I keep doing this.
Lol, like this one. You understood.
Correct. and it's going in this direction.
Correct.
DNA is being made from 5 prime to 3 prime. Yeah, now it's correct. Yeah. So the top strand is the template on the basis of which this RNA is being made. Okay. So it is called the template strand. Do you understand the template वाला concept?
Yeah. It's using
यहाँ पे
that to
DNA
make
कैसे
the
बन रहा था? The polymerase was reading कि इसके अंदर कौन सा बेस है and उसका complementary बेस को वो ला रहा था conservation. Correct. Similar thing is happening here. Like the polymerase is reading कि यहाँ पर कौन सा बेस है. अच्छा यहाँ पर T है, so I'll give you. And
Right. And it's just that the other strand is just chilling, right? It's not doing it.
The other strand is just chilling. It's vibing while the other gets replicated. It's like, I'm cool. I have to do no work. Easy life.
Right.
I'm in CSE. Yeah. No, RNA and DNA don't bond. U and A will bond, first of all, and C and G will not, like C and G will bond. The thing is that your OH group is the real reason. Because of the additional OH group, its structural chemistry is different. The spacing is more. So it doesn't align at all for it to form that double-stranded shape. DNA में this thing, yeah.
No, I think I got your question because we study in ITB that you can have triple-stranded DNA and four-stranded DNA, right? So you're like, why doesn't this form like triple-stranded DNA, like two strands of DNA,
with
one
RNA
strand of
and
RNA?
Just we're talking about something like ये DNA strand है and ये RNA strand है. नहीं हो सकता, मैं बताता हूँ क्यों. And even triple stranded नहीं हो सकता because of the same reason. So look at this. This is DNA and this is RNA का thing. What is the difference? This OH group, right? Don't you think this OH group will cause some kind of interaction which will change?
Oh, yeah, I get it.
So यहाँ पे इसलिए था because this it was sort of aligning because the distances were the same. Alignment was maintained here.
Yeah, one side distance is the same, the other side it is.
So suppose नहीं होता deoxy right? Actually length ज़्यादा होता है RNA का. So suppose तुम्हारा AGC sort of thing है. T मान लो, ठीक है. इसके corresponding RNA बनेगा तो A long length and it will have somewhere around here. It will have U. Then long length. You're seeing like वो match ही नहीं होगा. Like how will it form because when it's not even having the same strand. And these are rigid bonds, right? Unlike इन दोनों के बीच में hydrogen bonding हो रहा है. These are covalent bonds. You cannot bend covalent bonds for hydrogen bonds.
Now I know his next question. Then how is it replicating with the perfect sequence at so much distance? Because I have the same question.
With?
So like when it's finding its complement, right, from the DNA strand, but the RNA strand is longer. So technically the polymerase should be moving at different distances to attach one here and read one from there.
Arey bro, polymerase is only attaching to your DNA. That is something that you're forgetting again and again. If polymerase is attached to the DNA, it is reading क्या है DNA में.
Oh wait, so what is attaching to the RNA?
Suppose you are the polymerase, ठीक है? You have an assistant. Let's call him Chotu. Chotu will be a black guy because black guys are criminals. Sorry. Subtle racism. So yeah, so you have a bunch of orders that you have to fulfill. ठीक है? You are working this. Arihant is working in Amazon as a delivery boy for some reason. Manager. Okay. No. This is Arihant. No. It is Arihant. He has to like give orders. Orders वाला डब्बा यहाँ पर है, ठीक है? So एक बॉक्स आया. Arihant will first start with this box. He called Chotu for Chotu like order number 1, like 1 वाले का box ला. So he'll bring order number 1 का. Then Arihant will move to order number 2 and he'll like, hey Chotu order number 2 का ला. Or Chotu will bring. And so basically A T G C A T मान लो ये है. And छोटू जो boxes ला रहा है वो तुम्हारा corresponding है. So it's like T का box बन गया. अब A का box बन गया. It doesn't matter whether these are aligning or not because Arihant is the one who's making the orders. Arihant has to be aligned with this, but Arihant doesn't like he's just producing this, right? He doesn't care. ये align, ये दोनों align हो रहे हैं कि नहीं.
Oh, nice freedoms here.
Yeah.
So wait, so technically if you look at it, so there is the DNA polymerase is only used for reading from the template strand, and there is some sort
Yes.
of other some other entity that we don't know yet that attaches the nucleotides to the RNA strand, and this is like the messenger. That's it.
Other entity which is no, the DNA polymerase is doing the attachment. There are other entities that bring it towards the DNA polymerase.
Okay.
छोटू कुछ काम नहीं कर रहा. छोटू बस तुम्हें वेयर हाउस से चीजें ले आ रहा है.
Okay.
You are like you reach A, you are like छोटू टी कला. So he'll go to the warehouse where everything is stored. Then he'll bring out tea and you'll place it. Then you go to
Yeah,
T.
see the thing
You're
is,
like,
if it has to read from the DNA template,
sorry, this one is-
and if it has to read from the DNA template and work with the RNA at the same spot, that's not possible, right? So
Bro,
reading
RNA
is
is being
done.
produced as a string. It doesn't care about the RNA.
Yeah. It will just give the order that this is what has to come next, and something will come and
Huh.
attach there.
Yeah. It will get the raw material.
Good.
It is doing the attachment, all of it by itself. It just doesn't affect the order, like in the sense that just think of it this
I
way.
got that. So yeah, see the thing was,
Suppose ये डब्बे काफी uniformly placed है ना, like with almost equal distance. My entire premise was that RNA is going to be in different sizes, right? फर्क नहीं पड़ता. Like A के corresponding तुम्हारा डब्बा एक यहाँ पर है, दूसरा यहाँ पर है, तीसरा यहाँ पर है. Doesn't matter as long as you're getting the order done.
right.
How does it matter? Like as long as you're reading the order and doing all of that, what RNA you're producing doesn't matter, right?
So yeah, like I'll tell you why I had this confusion. I was thinking of it as literally a human working one hand touching something, feeling what it is, and then trying to have its complement on the other hand. But the thing is, the other hand has to reach way far ahead because of the length of whatever you're working with, that being the RNA. And the right hand side or the other side, he'll have to reach out far lesser than what he has to do on the like complement side, right?
Bro, ये
So that is
complicated
how I was
stuff.
imagining it.
Just think of
Yeah,
it as conveyor belt.
Yeah, I
Makes
get it.
sense.
हाँ.
Conveyor belt में यहाँ पर uniformly he has another conveyor belt here for the RNA.
Yeah, we just assume those things are moving at different speeds. That's all.
They are just moving in different speeds.
Yeah, I got it now. Yeah.
Yeah, that's it.
Okay, quite a lot happened. don't assume I'm not here. Don't carry on. I'll try to pick up.
Yeah. Yeah, so
Yeah.
so I did like intro to biostarting.
Yeah, cool.
I mean, sorry, intro to bioinformatics.
Bioinformatics I started. You finished ITB.
Not finished. We are almost there.
Yeah.
I am slow, as you would have realized by now.
I mean, we
Yes,
distracted
of course.
ourselves with operating systems, Windows 11, and all the other stuff for a
Yeah,
long
yeah,
time in
yeah.
between.
So remember this structure, okay? Like Chotu and because this structure is going to come in place again and again, just like your other structures are also coming into place, like DNA or eyesight to another concept and your thing. This
Mhm.
concept will also come into place.
Hmm.
So first, let me delete यहाँ का thing. I should actually put it in a different page. Let it stay here.
Lol.
So this is how replication is happening, right? I'll just redraw the DNA strand. I was trying to be lazy, ended up doing more work. So you have two DNA strands, and you have a polymerase, which is polymerizing what RNA this time,
Hmm.
polymerizing RNA on one string. Yes. See, I told you nomenclature in bio is very simple.
Yeah. So I'm guessing this would be DNA dependent RNA polymer.
Yes, DNA, perfect. DNA dependent.
Nice. It needs DNA, but it's doing
RNA.
RNA stuff.
Wow.
Yes, polymerase. GG. And this strand is called template strand. इस पे replication process हो रहा है, replication कह रहा हूँ, transcription process हो रहा है. That is called the template strand. And the strand which does no work is इसका नाम में हम लोग वापस आएंगे, ठीक है? Right, we come to that. Bad ass होने वाला है, like the sense that you remember you made a brilliant observation that नीचे वाले का same is आ रहा है, बस T छोड़ के U आ रहा है.
Hmm.
So in other words, RNA का पूरा कोड नीचे वाले में.
Hmm.
नीचे वाले मिलते ही तुम्हें RNA का कोड पता है.
Hmm.
So the one which is just chilling around is called coding strand
Oh, nice.
because it has the RNA का code. RNA का code with T and U वाला flip. So this actually doesn't code for anything. So it's a bad name, but it is the code. So yeah, I
It's, yeah,
suppose.
it's liquid.
Coding strand gives you questions क्या आ सकते हैं coding strand से कैसे तुम RNA ढूंढ सकते हो. You just replace T with U. Template strand से कैसे RNA बना सकते हो. You will have to do reverse complement and then flip T with U. Does it make sense?
Yeah.
Yeah. Because DNA वाला same concept है, reverse complement बनेगा, but this time it's RNA. So you have to do T replaced by U. Cool.
Right.
So that is the entire concept of transcription. Everything is same, right? You have an origin of replication. Jagah, now you have initiator site. Instead of RNA like DNA dependent DNA polymerase, you have DNA dependent RNA polymerase. Same reverse complement हो रहा है यहाँ पर. And because एक ही में हो रहा है, so we directly see that it's just a flip. Like, उसमें भी यही हो रहा था ना, like we got the two children, this one and the other child, like other
Yeah,
child,
they were exactly the same.
they are exactly the same. It's the same thing except अब RNA है तो तुम्हारा T की जगह U हो जाएगा बस. That's it.
Hmm.
So with that, transcription is also over. Now, like the reason why I saved our Arihant and Chotu wala scenario, translation. Now, before translation se pehle, there is some pre-translation processes that I think we should discuss. So, I said RNA banta hai, but like as Arihant rightly pointed out in between, like he remembered. I'm very glad he remembered. कि RNA multiple तरीके का होता है. Like you have most common is mRNA. We always seem to discuss
Right, that
mRNA.
was message RNA.
Messenger RNA. Now
Then there was
messenger
tRNA.
RNA
I
and
remember it.
tRNA. Transfer RNA. I'll give you a clue towards the third one. What are ribosomes made of?
40s and 30s stuff, or 40s, 70s.
Like those are the like for 40s and 30 like it's actually depends on what organism, but these are just
Yeah.
basically units of where they lie in the what do you call what was that thing, the centrifugation. No, it is rRNA, ribosomal
I don't think
RNA.
I've heard.
Ribosomes are made of RNA.
Interesting.
So let's go back to our central dogma and RNA world hypothesis. So I told you like early life was all RNA. Like you had double-stranded RNAs, which would make single-stranded RNA, then you make proteins. Cool. Now you realize that process of making proteins also has RNA. The machinery is made of RNA. So essentially, the bulk of genetic material transfer is being done by RNA.
Right.
It is bringing in amino acids. It is like actually bringing together the amino acids. Hence, this is called RNA world. This is how early life started, with only RNA. सारा काम RNA कर रहा है ना, मतलब it is what is doing tRNA, mRNA, rRNA सब कुछ खुद कर रहा है. So everything that is required for transcription is being done by RNA. Anyway, like this will be slightly more clear once I explain transcription, I suppose. Yeah, I'll revisit that once we are done. So अब mRNA जो होता है, what happens is that if you are a bacteria, then it's fine. mRNA बन गया, तुम्हारा directly translate हो जाएगा. But if you are a human, I hope you are humans. Like it's slightly more complicated. You have coding and non-coding regions. Here, do you remember the names, Arihant and Shreyas?
Yeah, I remember coding, non-coding. The coding regions make using the coding regions you can make proteins through the entire process, but using non-coding regions they don't exactly code any particular protein.
Yeah. So what has to be done to them?
You kind of have to cut and take only the coding regions. Right? Wait, I'll tell you the name.
Yes.
I forgot the terms for this, but
yeah. So it has coding and non-coding regions. So the coding regions, the non-coding regions actually occur in between the coding regions. Because they are in between, they are called introns.
introns and exons, yeah.
And because these are on the exterior, they're called exons. Nomenclature in biology has some nice sort of tricks, like mRNA is messenger RNA, tRNA, transfer RNA. Like if you get hold of that naming technique, like just the way you get DNA dependent RNA polymerase, like once you get
hold of it seems very logical naming.
Yeah, the naming is very logical once you get. I mean, that did happen, right? Okazaki fragments.
yeah, that also happened.
Yeah. So, I mean, that also happens, but like lots of stuff has logic to it. That's something that people sadly forget. Like they think that biology is all about memorization. So, introns and exons. Now, as Aryan rightly pointed out, like these non-coding regions are useless. As far as protein making is concerned, these non-coding regions actually add. No, introns are the non-coding regions. Exons are the coding regions. So, like they are useless when it comes to protein making. They are not useless in general. They are very important for structural stability of the entire like RNA molecule. In fact, not the RNA molecule. They are actually there in the DNA sequence itself for structural stability. Their structural stability is actually in the DNA sequence. So this RNA is coming from DNA, right? So the DNA corresponding to the introns are there for structural reasons. But now once we are going to produce proteins, now this structural reason is no longer valid and this is just useless for us. तो ऐसे निकालना है. So that process of nikalifying is called splicing.
Right.
So कैसे निकालते हैं? how it happens is that this thing sort of coils in this in this manner. So it basically looks like and it gets cut like this. So you end up having one nice beautiful exon wala thing. That's not it. Like इसके end में there's something known as poly tail that has to be added. So this has been done. Now suppose this is the 5 prime and 3 prime. So once splicing has been done, even now it's not perfect. So what you do is that you add two stuff like to the mRNA. That is, one is G cap. G cap is basically you put a guanine group in front. You just add a G just for the sake of it. And towards the end you add A करके. This will go to like around hundreds of bases. Just A. So no, I'll come to the reason. So this could go from tens to hundreds depending on what organism. So like,
So wait, I mean, you know that we had to do that Wuhan genome, right? Even that thing had like crazy number of A's at the end. Is this the reason?
Wuhan, it's an RNA virus, right? So
Yeah.
RNA virus, when it's been reverse transcripted, it will always have poly A tails, right? So एक का complement करके आ रहा है. That's it.
Okay.
So why this A is because of this machine. Like, say you think of this, हम लोग protein हर दिन बनाते हैं, correct? Which means your gene is being expressed every day. Doesn't it mean that your RNA molecules are randomly floating around everywhere? Won't it be bad? Where is all this wastage going? That is one. Second is that if RNA molecule is always there, won't it continuously keep on making proteins again and again and again and again and again? Right.
Right.
What is there to stop protein expression? So the thing is, your RNA doesn't stay in your body for a long time. Like RNA is continuously being attacked by enzymes which are cutting it down. Guess what these enzymes which are cutting it down are called?
RNA. Oh. Huh.
So restriction enzymes are actually cutting it. They are degrading RNA molecule at every time. So the purpose of poly A tail and this G cap is for directionally guiding it so that RNA what do you call your ribosomes can recognize it. This poly A tail is to prevent early cutting by restriction enzymes.
Oh, okay.
So restriction enzymes. First question you could ask is that poly A tail. How does it affect restriction enzymes at all? Can't they just directly cut? The thing is that restriction enzymes are released by
some direction, right?
it is released by some factors. So
It
what
has to
happens
find
is
that
that
restriction
kind of
site, right?
The
It
thing
just
is that
takes longer
restriction
to find
enzymes
it. Huh.
that we generally study over palindromic sequences, right? But in nature, restriction enzymes cut everything. Like palindrome is not a requirement for restriction enzyme. So if you search list of restriction enzymes, you will find that it has non-palindromic sequences also.
Okay.
Like it has wildcard entries, it has stuff which are not palindromic. Okay, these are again this one. Yeah. So, like non-palindromic sequences. So the purpose is basically to digest restriction enzyme. Why are they present in nature? They are there to digest what do you call your RNA molecules that are just lying there. So and these are also released by other factors. There are other chemicals which also cut it, other than restriction enzymes. Like restriction enzymes is of interest to us because we use it, but doesn't mean that nature doesn't use other stuff. So this poly A tail prevents it from being cut early. Like the probability of cuts happening becomes much lower because the length of poly A tail is long enough to prevent it from getting cut before expression. Correct, correct. Huh. Individual basis will be still there. Huh. So again, as I said, restriction enzymes are not the only thing. They cut it, your smaller strands, cut it again, there are much smaller strands and so on and so on. It's like digestion process, right? Digestion में होता क्या? Like suppose you are digesting proteins. तुमने अंडा खाया, तुम्हारा protein digest होगा. Protein is going to be a huge amino acid chain. In fact, it is going to be some kind of folded amino acid chain. So one protein will break, like it will unravel this thing. Then random cuts will be added. Then again, random cuts will be added depending on what protein it is. And then again and again till you eventually have an amino acid sequence that you can digest. CNC is restriction enzymes में होता है. It can't code anything, and in fact, it will keep on cutting till you only have the bases. Like in the sense that sugar with base, that is. ये बचेगा. Of RNA मतलब. Yeah. So that is the role of restriction enzyme, and this poly A tail is called poly A tail. This is G cap. Poly A tail prevents it from getting cut preemptively by restriction enzyme. You don't want it to cut too before it's expressed. So this sort of processing is done before your transcription process. Sriram, any doubts? Because Sriram came late and I feel sort of guilty for starting without him.
No.
Sriram?
No.
You
No,
must feel
no
like a
doubt.
professor.
Okay.
No.
I don't feel like a professor. I feel like I should have waited for Sriram.
No.
Because like yeah,
Wait, what?
because this RNA sort of thing, like I think Sriram would have enjoyed. Like double
Yeah.
stranded RNA.
Oh, you'll upload it on room 333.
Yeah, we were talking about operating system.
Yeah, OS and deleting BIOSes.
I can't imagine
Yeah.
Hrishi talking about OS. Interesting.
Oh bhai.
If we get back into it, then we might not finish it tonight. So we'll
What? Hrishi?
Winter vacation, bro. Winter vacation. Application, yeah.
That is an OS.
Basically Linux, bro. Like, okay, I work with OS in the sense that non-theoretically, practically, I Google that stuff and I do.
I thought you were doing Ramsey level OS. I'm like, you did that in summer?
Bro, come on. I do
Exactly.
the boring stuff, bro.
Hey, oh. Tell us to go over, bro.
I mean, I think Arju has told him to. I'm fine with system administration work. Like, it's actually interesting. OS, no.
Yeah.
So now we come to transcription. I forgot numbering also. 3. It's the third step, right? Translation, sorry. So translation is what? Your RNA to protein. And we had this entire discussion about translation logic here, like we're going from nucleic acids to amino acids. So it's like a different language altogether. Another way you can think of it is that it's going from code to actual expression. It's getting translated into something as an outcome. So like that's how translation works anyway. Like that is the basic idea. Now before we talk about translation, let's go back to Arihant. Okay. So what Arihant was doing is that he's getting like orders ready, right? And he has this Chotu who is helping him out. Chotu will take out orders from this warehouse of theirs. Warehouse में सारा सामान है जो भी order पे होगा, and Arihant will put it in the outgoing conveyor. This is basically my output conveyor and this is my input conveyor. इस direction में आ रहा है, ठीक? Now मेरा Arihant जो है, मेरा Arihant लम्हा, Arihant जो है, like he was doing it one by one पहले, but the company policy changed and he has to read 3 at a time, like 3 boxes at a time. And so like 3 boxes are all 3 orders are basically going to the same place, same location kind of thing. He reads these 3 boxes and then he calls for Chotu to bring whatever is corresponding to these 3 orders, packs them into one huge box and sends it. Then he reads the next 3 and he's starting to work like that. Hello?
Yeah.
So this is exactly our amino acid wala model. So what happens is that your Arihant is your ribosome.
I see.
This is your RNA.
Nice.
With 3 prime to 5 prime. Wait, my bad.
Reverse.
5 prime to 3 prime. Yeah. I mess up the numbering every single time, bro. it's irritating. So this is your ribosome. What was our ribosome? Our RNA.
Right. How?
And like ribosome is calling for Chotu for getting him the stuff he needs, right? So that he can put in the box. So and Chotu will go to the warehouse and bring the stuff and so on. This Chotu, who does the entire work of transferring from the warehouse to Arihant, who will actually make the order, is you're basically transferring it from the he's basically transferring it from the warehouse to Arihant, right? His work is of transferring. He is the tRNA. And what is this warehouse? It is basically all of this is happening within what? Nucleus. No, wait. This is happening in your SCRs and RER. That is soft endoplasmic reticulum and rough endoplasmic reticulum. Ribosomes are more abundant in rough, that's why it gives a rough-like appearance. And soft has less ribosomes, so it has soft appearance. So most of your production is happening in RER, and like both SCR and RER are responsible for protein production. But your amino acids are not in your SCR or RER. They are produced by the Golgi bodies and sent to the cytoplasm. So to give context, like this is how cell is double membrane. This just assume that this is a beautiful cell diagram.
Yeah, that's right.
Yeah. So it is double. I feel guilty now.
So it's fed.
I'll make a stopgap thing, but better discussion. Yeah. So this is your cell membrane, which is a double membrane thing with pores in between. Similarly, you have another double layered membrane, which is around your nucleus, which is called nuclear membrane. You have cytoplasm, and you have RERs and SCR, which are sort of attached to your nuclear membrane, all around the nuclear membrane actually. And you have other bodies, including mitochondria would be here, and you have something known as Golgi bodies, which is like concave, T-shaped sort of stuff. I think I should show an image of.
I mean, we know what it looks like. I remember Golgi body. It looks like Wi-Fi.
What just happened? Oh yeah, nothing. So you know what it looks like, right? So
Yeah.
Golgi bodies are the ones which release the free amino acids. Amino acids are obtained from where half of. So few of your amino acids are produced by the body. Those are produced by Golgi body. But some of the amino acids.
Oh, these
not
are
produced by the
essentials
body.
and non-essential.
Yes, essentials are the ones which are not produced by the body. So it's essential that you consume them. That is how the word essential comes from.
Okay.
Essential, you don't.
Wait, what? Repeat that?
Essential is
Essential
something that
proteins.
your body requests from outside. Like you need to consume it. It's essential for your body. You don't make it. Non-essential is something your body makes.
Yes. It is made by the Golgi bodies. Yeah. You don't need to
Cool.
get it from food directly. Essential are those which you need to get from outside sources because your body can produce, and non-essential are stuff that you can make. So, well, because you can make, why would you get it from outside? So it's not essential to your consumption. So, I'm not like I don't remember the exact number. I think 21 कुछ था ना? No, there are total 20. You are right. 20 amino acids, out of which 1 second. There are 21 amino acids, and one of them is not like considered to be an amino acid because it's not part of the standard genetic code. It's only produced by like bacteria. It's selenocytostine. I don't know whether I'm pronouncing it correctly, but no, like no. All 20 are not made by our body. These 20 are 21 if you look at the exchange case, but let's assume for 20 in this discussion. Let's not go into complications. I should not have mentioned 21 at all. Yeah, so 20 amino acids. All of these 20 amino acids are the total number of amino acids any organism can make. Like it doesn't matter human hai, koi hai. These are the possible total number of possible amino acids that can be made. But like there are some amino acids that humans can make. This includes, I don't know the exact number. I'll have to Google it up.
Yeah, exactly. But among these 20, there are some we can make some like that.
Okay, so 9 amino acids are essential, and 11 are non-essential. So basically, 9 are not produced by us; they have to be obtained from outside sources, and like rest 11 are made by us, so they don't have to be obtained from outside sources.
Nice.
Yeah, cool. So there are essential and non-essential amino acids which are sort of outside our description. But anyway, the reason why I mentioned all of this is that Golgi bodies is producing it and it's leaving it in this cytoplasm. Cool. Now your amino acid is in the cytoplasm, so reaction चल रहा है within this, right? Ribosome, like SCR and RR, endoplasmic reticulum. What will, like, why would randomly amino acids go there? They won't, right? There needs to be something that pulls it inside. That is what tRNA does. So cytoplasm is your warehouse.
I see.
Cool.
So wait, 1 second. The DNA replication and transcription, right? All this is to do with DNA and RNA, which is present only in the nucleus. So those two processes take
Yes.
place completely in the nucleus.
Yes. So I should have mentioned this before actually. Replication, transcription, both are in the DNA because nucleic acids are also in the DNA. Even the transfer that is happening, it's a short transfer because everything is within the nucleic. This is called nucleoplasm. The plasma within the nucleus is called nucleoplasm. So everything is within the nucleoplasm. So you mentioned
Okay.
like when I mentioned that factors are bringing it, it is much less of a job for both a DNA replication and RNA replication than it is for protein, because protein may it is very far away, like it's a complete entirely different body which has to
Right.
bring
Right.
it in.
Hmm.
Yeah. So that is basically the role of tRNA. tRNA basically collects cytoplasm, like collects amino acids from the cytoplasm. Now tRNA मैंने like अब मुझे थोड़ा केस चेंज करना पड़ेगा to make it the actual case. Arihant doesn't have one assistant, he has multiple chotus. ठीक है. And because like you want to save up on time in this order, so you tell chotus कि तुम सब लोग एक एक बॉक्स पकड़ के आ जाओ. मतलब one will hold any random amino acid of your choice. And random नहीं, मतलब bring all possible.
It's like getting
No.
it in the stock or something.
Yeah, correct. So there will be lots of chotus who are already holding boxes with amino acids. अरिहंत को जो भी ऑर्डर चाहिए वो अचानक ले लेगा उनसे. लेने के बाद छोटू विल गो बैक बिकॉज उसे नहीं पता कि कब अरिहंत को चाहिए. Now there will be multiple chotus with the same box also. Like it is so that अगर अरिहंत को सेम बॉक्स दो बार चाहिए होगा, so he can provide that.
I see.
Cool. So this is exactly how transcription works. So how it looks like is very similar to whatever we have covered. Actually, this conveyor belt thing is exactly how it looks like also. You have Arihant who's doing this. He has another conveyor belt of proteins being produced. And he has chotus, which are tRNA. tRNA has an L shape, but like it sort of looks like a weird L. And it has a binding site, which is basically the amino acid binding site, and it has an anticodon loop. So anticodon loop of each tRNA has like for example methionine का मान लो. Methionine इसलिए consider कर रहा हूँ because there is only one like sequence that is coding for it, right? Which is AUG, and it's the start sequence also. So methionine का sequence क्या है तुम्हारा? AUG है. If I'm reading AUG like this, so my conveyor belt is moving in this direction. Like because
Okay, so you mean the one that is going to call, which the mRNA has, should be the complement of AUG, right?
yes. So this anticodon loop of the tRNA that the rRNA is going to call should have complement of AUG. So it should have UAC, and this will hold methionine.
Okay.
So what holds methionine is this tRNA with the anticodon loop. So what happens is that this all of these tRNAs are here, right? rRNA, what it does is that it forces them together. So this tRNA basically ends up like this. So I should have made rRNA a bit better in this case.
Yeah, I mean, we got it.
No, like I'm very afraid that I'm not making certain stuff clear. Why am I so dumb?
Okay.
Cool.
Mm-hmm.
So tRNA, sort of binds with the rRNA. Like it's a huge complex mechanism. I'm not going into it. And what it does is that this ribosomal RNA is already holding whatever protein it has already generated, like sequence it has generated. It just adds methionine. Next sequence, suppose मान लो A है, ये बाहर जाएगा ऐसे, another thing will come, like this sort of tRNA will
the
come.
same figure is going to.
Man, I'm bad at drawing. F.
So I mean, it's pretty good. We understood everything.
So this is LU, right? I might be wrong with this, but like bear with me.
Yes,
Point
like,
is like-
conceptually we got it.
हाँ, तो ये आएगा and ये आने के बाद like same thing will happen. And this goes out, it will be basically this structure without this methionine.
Right.
This
And this would have moved by 3 spaces.
this would have moved by 3 spaces. See, you see how the anticodon loop is also working, right? It's a twisting motion, so that is what is actually pulling also. The initial part is actually supplied by the bottom end of the. It has a heavier end and it has a short, like lighter end. When I say 80s and 50s, and together they make 100s. First thing you would ask is that 50 plus 50, how is it making 100?
Yeah.
Right.
I know it doesn't exactly work.
Yeah, the reason is that this unit is sort of not what you think it is. It's something called Swerberg unit. So basically it is I'm not sure of the pronunciation anyway, right? So it's basically when you do centrifugation, it is the measure of how far from the center you are.
Oh,
80s is something that is very far, 50s is this. But together, they need not have the same density, right? 80s might be bigger. It's becoming 100s, it means that 50s is actually more in quantity than 80s.
Right.
Your centrifugation is dependent on density, right?
It's like a weighted mean kind of thing.
Yeah, it's a weighted mean kind of thing. It tells you what proportion of mass is actually there. It doesn't tell you anything else. So 80s and 50s. 80s and 50s is basically your ribosomal units. The bigger unit is 80s and 50s. It's the measure of their density. That's it. Yeah. Not important. Like this was there in ITB and like, so I re-mentioned it. Yeah. So this is how your new amino acid is created. Cool. Is this clear? Yeah. Now, like I will show you the like. This is bio-NCRT, by the way.
I've never seen it.
So one second, I'll show you amino acid like transcription or thing. They have made it like this, but the actual shape is slightly L-shaped. Yeah. So does this clear it better? The image, that is.
Yeah, I mean, I kind of got it. This image looks more complicated than what you do.
Yeah. Just think of Arihant and his
And my
like,
choice.
yeah, bro. Look at this. I think this should clear it. So tRNA comes in, binds. The older tRNA goes off. I'm sorry. Oh no. This like this green thing is ribosome, bro. rRNA is this ribosome is not a protein structure, it's an RNA structure. No, rRNA are basically RNA strings, okay, which coil up in weird shapes to form this upper wala chunk and niche wala chunk. Similar to amino acid folding, right? Nothing complicated there. So that is ribosome, and so like here I was not able to show कि कैसे hold कर रहा है, but here they have shown clearly कि कैसे holding process भी हो रहा है. Like this is the string that has been generated is held, the next one is attached, and new one is brought in, and the old one is sent out. Cool. Yeah, so that is basically our translation wala thing. And how the translation happens is that you need an AUG to start, correct? And after that your reading frame is fixed, and then you start reading in pairs of 3. Cool. So this is the table of various codons. You don't really need it, but just remember that there are, how many possibilities do you have of 3? 4:3, right? 64. But if you look at this, how many amino acids do we have? Like we discussed this right now, right? कितने amino acids हैं? Amino acids कितने हैं, bro?
20 amino acids.
20 amino acids. So you have 20 amino acids, and you have 64 possibilities.
Yeah, but most of them
It is very
go for the
obvious
same
by
thing.
pigeonhole principle that
Yeah.
principle, bro. Like there should be more than one for. I mean, at least one amino acid should be there so that you have multiple in it. Now this brings in this weird concept of degeneracy. Degeneracy. We say like amino acids are degenerate. Amino acids are degenerate. What degenerate means is this. If I give you a DNA, what its parent DNA was, correct? Hello?
Wait, what? If you give us a DNA?
If I give you a DNA strand, okay, you can directly tell me what its parent was. Parent DNA was.
Right.
What its template would have been. So template would have been. Cool. Similarly, if I give you an RNA strand, तो भी तुम बता पाओगे what the parent was. What if I give you a protein? Exactly, because repetition, for the first time you cannot comment on what the parent was. What is the implication of this? Two sequences which are not equal to each other and code for the same protein.
Right.
So you can get the same protein from two completely different possible sets of strings. So this means that backward thing is not possible. So it can be the same protein or same amino acid can be obtained by more than one codon. Right. That is the essential idea, right? So hence it is called degenerate, because it is, you cannot generate it, so it is degenerate. You cannot generate the RNA. It is, you cannot cut the, so yeah. And
yeah, exactly. That's like interesting gaming.
Yes. So AG has dual function that you already know, and like this code that we know, that is UUU, UAG. This is universal. Like no matter what organism you're looking at, AUG will always give you methionine. U will always give you PHE. What is PHE? Phenylalanine. Yeah. So it doesn't matter whether it's bacteria or human or I don't know, jellyfish or I don't know, money plant, Jagruti, anything. It doesn't matter. Everyone will code the same proteins for the same codon.
I see.
Huh. That is second and last thing that I have to like sort of cover before we reach biotechnology is the idea of reading frame. So initially the string doesn't care कि कैसे read करें. It's reading characters one by one. But once it finds AUG, it binds onto it. The ribosome binds onto it and starts reading in pairs of 3.
Mm-hmm. Yeah.
So why is this reading frame important? Like NCRT has a very good example. I think मैंने पिछले दिन शेष को भी ये बोला था. Suppose this statement is there. Ram has red cap. ठीक है. Suppose by some mutation गलती से insertion आ गया. Now you see how the reading frame has been.
Yeah.
आप पढ़ ही नहीं सकते. कुछ गलत हो गया. अगर मैंने सपोज AM से स्टार्ट किया था तो I would have got ASR, ADC, AP. So the way I read is like sort of dependent upon how I start. So
Right.
you might find मतलब what it basically means that if you have an arbitrary string T नहीं होगा ना मतलब G C so on and somewhere in the string you find ACG. There's an AUG somewhere here. Let's assume this part is being encoded. मान लो okay AUG start हुआ था. You cannot say for certain that ACG corresponding जो भी है वो express होगा कि नहीं. It will only express if it's within the reading frame. ऐसा हो सकता है reading frame looks like this.
Right.
Instead
Right.
of, or it can be something of this order also, right? मतलब
Correct.
and whatever is next. So just by looking at 3 section, you cannot directly say what it is coding for. It might be part of 2 separate amino acid codons. So that is the importance of reading frame. And reading frame is what causes most of the mutations.
I see. Huh.
Yeah. Can I take one last thing, which is not a huge thing, that is that the region before AUG, first AUG, and the region after stop codon to the next AUG, but like more generally you can say region after stop codon are untranslated, right? It doesn't do anything. So that is called UTR, untranslated region. Bus. So intro to bio part is more. Yeah, even my throat is sort of going so.
Yeah, I'll go get
Yes,
some midnight snack.
Yes, Rishi, I will allow you to take up.
Okay, sir.